So this outer radiating cells, these are corona radiata. Okay, so initial phase is it penetrates the corona radiata. Then second step, it penetrates the zona pellucida. Okay, and third, it penetrates finally the membrane. Okay, cytoplasm, it enters the cytoplasm finally. So for this process, okay, so you can see, so for this process, okay, there should be certain changes in the sperm. It should be capable, what we call capacitation. And here you can see in the head of the sperm, so there are certain acrosomal enzymes. Okay, so you can see the enzymes. And what is acrosomal reaction? It releases certain enzymes that help in penetration. Okay, that helps in further penetration. These are the proteolytic enzymes. So it will make a pathway, okay, where the sperm can go. Okay, and it will penetrate, and finally it releases the sperm in the cytoplasm of ovum. Okay, so this is about fertilization. So sperm penetrates the corona radiata, then zona pellucida. Okay, and after one of the sperm it penetrates the zona pellucida, there will be zona reaction, because to prevent the polyspermy. Every sperm, once it fertilizes, then there shouldn't be, others should be blocked. You know, also block pass any. So you can see here, okay, these are all are blocked. Why? Because of zona reaction. There's certain reaction, okay, that inhibits the further penetration of spermatozoa, what we call is a zona reaction, and then there will be fertilization. Okay, and before fertilization, what will be the changes in the female gamete? Okay, in the ovum, the secondary oocyte, it complete the meiotic division. Okay, so after fertilization, there will be male pronucleus and female pronucleus. Okay, then they start dividing. Okay, and that stage is called a cleavage. Okay, so let's revise again. So let's see in this female genital tract. Okay, so once the ovum is mature, so this is secondary oocyte. So graphene follicle, this is the secondary oocyte. Okay, now it get released. Okay, it released after LH surge, and once it is released, it is arrested in the metaphase. So you can see here this is arrested in the metaphase. Okay, and the sperm will penetrate. Only one get that opportunity. Okay, because to prevent the polyspermy, there will be changes in the zona reaction. So this red lining, okay, this is the concentric red lining, this is the zona pellucida. Okay, it prevents the further penetration of other sperm, and now it forms the this specific. So once the sperm has entered the cytoplasm of ovum, it completes the second meiotic division. So finally the ovum matures only after fertilization, then it start the division. Okay, that we call cleavage. Okay, that means just you cut it. Okay, because of zona pellucida, the size won't be any difference in the size. Okay, just 1, 2 cells, 3 cells, 8 cells, 16 cell, like that. Okay, it's just like a cutting a pie. Okay, so you can see 6 cell stage. No, sorry, so it can be, so in 30 hours you can see 2 to 4 cell stage, then 8 cell stage, then 16 cell stage. Okay, mathematically it will divide, and so 16 cell stage, what we call, it's a morula, and this morula, it's like a ball of cells, it moves into the cavity on 5th day. Okay, for and after 5th day, now in the morula, these cells, okay, it start certain fluid to get inside. Okay, it suck the fluid from the uterine cavity, and this stage is called blastocyst. So blastocyst is the one that get implanted. Okay, so let's discuss in a sequence again. So what is the main result of fertilization? So basically it's a restoration of diploid number of chromosomes, as we know from. Okay, so this is, let's say this is spermatogonia, this is 2n diploid, and this is oogonia, let's say it's 2n, but it gives to 4. Okay, 4 sperms, all these are haploid. No, all these are haploid, and from 1 oogonia, it gives rise to 3 polar body. Okay, 3 polar body and 1 ovum. Okay, and this is also haploid. And when there is a fusion of oocyte and sperm, okay, when there is the diffusion, okay, of oocyte and sperm, N plus N, it will restore the chromosome. So main result of fertilization is there will be restoration of, okay, so diploid number of chromosome. Okay, another is sex determination. Okay, so in X chromosome, sorry, in sperm, it can be X sperm or it can be Y sperm. But what about the ovum? It's always X. Okay, it's always X. So it can be XX or it can be XY. Okay, so an X carrying sperm produce female XX embryo. Okay, and Y carrying sperm, it will carry XY male embryo. Okay, hence the chromosomal sex is determined at the time of fertilization, though it takes, okay, certain time to know phenotypically, okay, the gender of male or female. So it may take as late as 12 to 16, 12 to 14 weeks. Okay, but chromosomal sex determination occurs at the time of fertilization. Okay, so once there is fertilization, now there will be initiation of cleavage. Cleavage is just basically a mitotic cell division. Okay, so multiplication. Why we call it cleavage? There is no change in the size. Okay, in cleavage the size is fixed. Okay, there is just, it's like a cutting a pie. Okay, so the number of cells they divide, but they are in the fixed. It's like within a you can remember as a egg cell. Okay, so it's not cracked, but within the cell there is division. No increase in the cell size. Why? Because of the zona pellucida. It's because of zona pellucida. Okay, they cannot expand. Okay, and what about if there is no fertilization? Then the oocyte usually degenerates within 24 hours of ovulation, and the degenerated oocyte it is secondary oocyte. It's not a ovum. Okay, so am I clear so far? Am I audible? Okay, if there's no confusion, if there's anything, then you can text me or you can raise hand and ask. Okay, so again I will okay, so let's discuss about the cleavage. Okay, so let's see. So this is the zona pellucida. So once there is fertilization, okay, so this is within the ovum. Okay, what we call otid. Okay, otid or otid. Okay, and so these are the zona pellucida, and these are the polar body. Okay, so this is once fertilized, male and female pronucleus, they start division cleavage, what we call blastomeres. These cells are called blastomeres. Okay, they divide into two cells from 2 to 4, 4 to 8. Okay, till 8 they are very loosely packed, and in morula they are tightly packed cells, but within the cell. So within, it's like imagine a cells dividing within the egg, hence egg. Okay, you can imagine in that way. So there is no expansion. Why? It's because of zona pellucida. Remember, zona pellucida is like a outer shell of egg. Okay, so it doesn't give space for expanding. Okay, so basically it is a repeated mitotic division. Okay, cleavage is a basically repeated mitotic division of zygote, and these cells what we call blastomere. And when the cells proceed, okay, they divide and divide. So 16 cell stage what we call morula. This is the ball of cells. Okay, and approximately morula, it is forms approximately 3 days after the fertilization. Okay, and these compact cells, okay, so inside, okay, inside of the morula, okay, they are inner cell mass. They get divided, they get segregated. Okay, this is inner cell mass, and outside they are outer cell mass. And this outer cell mass, okay, they give rise to placenta. Okay, this becomes trophoblast, future placenta. Okay, and the inner cell mass, it gives rise to, yes, embryo. Okay, so when the morula enters the uterine cavity, yes, it enters around 4 days, and now, okay, there are certain changes. Okay, so in the zona pellucida, it becomes leaky. Okay, zona pellucida, it becomes leaky, and there is a certain amount of fluid enters the zona pellucida. Okay, this morula. Okay, and the cell they get segregated. Okay, and the cavity start formation. This cavity is called blastocystic cavity or blastocele. Okay, as as the fluid enters. So I will show you in the picture. Okay, so you can see. So in early blastocyst, okay, now in uterine cavity, it sucks the fluid and the fluid enters, okay, forming the cavity. Okay. And even in the cavity, look, here is the segregation of cells. The embryoblast, which is the future embryo, okay, they reside